This is Unit 22, Digestive System, Part D, Metabolism. This part is divided into two videos. This is the first part. It includes the liver, cell metabolism, nutrients, and measurements. The second part will include nutrient pools and states, blood glucose, regulation of feeding, and regulation of body temperature. Let's look at the liver first. What are its functions? One function is to process absorbed substances. Nutrients, for example, are processed in the liver. Carbohydrates are changed to glucose, if that has not already happened, or to glycogen as a storage form. Amino acids are made into proteins, and there are many other processes of absorbed materials. Drugs, for example. The liver will break down drugs and decrease the effectiveness of therapeutic drugs that are taken orally. The liver is a metabolic center for many different activities. Detoxification of nitrogenous wastes occur, including alcohol, poisons, and other ingested substances. Nitrogenous wastes include ammonia and urea. These result from the deamination of amino acids. You may remember deamination is the removal of the amino group from the amino acid. And as I'll probably say again, the body has a problem because it has no way of storing the excess amino acids. The liver is a manufacturing center. Bile, for example, is made by the liver. Bile is composed of water, electrolytes, bile salts, and bile pigments, as well as cholesterol and fats. So it's a fairly complex solution. Proteins and amino acids are manufactured by the liver. All of the blood proteins are produced there. Liver damage often leads to decreased plasma proteins, resulting in a decreased colloidal osmotic pressure. Red blood cells are made in the embryonic liver. By the way, if you ever have a test question that says, what is a function of the liver, you're safe to say manufacturing proteins, because that's a very common role. The liver is also a storage organ. It stores iron and copper, glycogen. It has about 100 grams of glycogen, which amounts to about 4 hours worth under maximum use. 60% of the glucose in an average meal is transformed into glycogen. Adipose tissue, notice, contains approximately 1 month's worth of energy. Vitamin A and vitamin D are two other forms that are stored in the liver. Regarding the blood supply to the liver has a dual blood supply. One is by way of the hepatic portal system. This system transports most of the nutrients that are absorbed to the liver. Blood travels from the GI tract through inferior and superior mesenteric veins into the hepatic portal vein to the liver. The hepatic portal vein supplies roughly 1,100 milliliters a minute and transports, as we said, almost all of the absorbed nutrients. There is a collateral circuit. From the stomach, it passes through various veins to the base of the esophagus and then by way of the azygous vein to the heart. Eventually, that circuit of blood returns to the liver through the hepatic artery. The hepatic artery is the other route. It supplies roughly 400 milliliters a minute. Think of that as supplying the nutrients and oxygen to the liver. Keep the liver cells alive. Lipids that are absorbed into the lymph system are also transported eventually by way of the hepatic artery. Blood exits the liver through the hepatic vein, which drains 1,500 milliliters a minute. Notice that's the total 11 plus 400. The blood is transported into the inferior vena cava and to the heart. Sometimes drugs are placed under the tongue of a patient. It's a sublingual administration. Why? What's the advantage? Well, is it absorbed faster? Perhaps. The patient cannot swallow? Well, that may be also. But mainly the drug will bypass the liver, so it's not broken down as fast. Sometimes the portal circuits will be obstructed. It may be a result of alcoholism or other liver diseases. It causes the pressure of the blood in the portal circuit to increase. This is portal hypertension. So it obstructs the flow through the liver, and the blood then has to find an alternate route. From the upper GI, the blood will flow through the esophageal veins to the heart in the collateral circuit we mentioned. The esophageal veins in the base of the esophagus are non-elastic and fragile, and they may rupture, leading to esophageal varices, bleeding. It's a possible life-threatening condition because you can't just put a Band-Aid on it. In the lower GI tract, there are also effects of obstruction. The normal blood flow is from the inferior mesenteric vein that drains the upper rectum and the distal colon by many different branches through the splenic vein to the hepatic portal vein and to the liver. The collateral circuit exists that allows blood to flow from other branches, including the hemorrhoidal veins of the rectum, to the heart. When portal pressure increases, blood flow through the liver decreases. The lateral flow through the hemorrhoidal veins increases. The hemorrhoidal veins rupture, leading to rectal bleeding, rectal varices. This is another consequence of long-term alcoholism. Sometimes drugs are placed in the rectum of a patient, rectal administration. Why would that be? What's the advantage? Again, not that it's absorbed faster, not that the patient is comatose, or these might be roles, but the drug will bypass the liver, so it's not broken down as fast. The veins of the distal rectum are not part of the hepatic portal system. Nutrients required by the tissues include minerals like sodium, potassium, calcium, iron, and so forth, vitamins, which are either fat or water soluble. Fat-soluble vitamins include vitamin A, D, E, and K. They don't have to be digested, but they're absorbed from my cells along with the fats. Water-soluble vitamins, on the other hand, they include most of the other vitamins. Most of them are absorbed as is. Vitamin B12, however, is needed for red blood cell production. Absorption requires an intrinsic factor. It's released from the stomach. It binds to B12, so the intrinsic factor B12 combination is then absorbed from the ileum. Decreases in B12 lead to pernicious anemia. It may result from stomach or ileum removal, because if something happens to the stomach, there's no intrinsic factor. If the ileum has been removed, there's no absorption of the B12. Nutrient pools refer to the total free nutrients in the body, including amino acids, carbohydrates, which are in the form of 100% glucose, and fats. They're in transport or waiting transport in the blood, interstitial fluid, and other extracellular fluids. Remember, the interstitial fluid is quite large, 3 times as much interstitial fluid as blood exists. From the nutrient pools, the nutrients may be put into storage, carbohydrates, for example, fats in the adipose tissue, the liver, which stores a lot of different substances. They may be used for energy, a topic we'll describe shortly. They may be used as building material. Some of the nutrients may be excreted, but this is very slight. Some from the cells and molecules are lost in the form of cells that slough off. Some are excreted in the urine. The details, as we said, of energy generation will be described. Cell metabolism. We need a little background for this. This is maybe in part a repeat of what we covered in the cell unit. So it may serve as a review. Cells require energy and building material. Where and how do they get energy? To answer, we need to look at several things. We need to look at ATP, which is the main source of energy that's required. We need to understand what oxidation-reduction reactions are all about. We need to know that there are two forms of cellular processes, two major forms, that generate the energy. One is cellular respiration, the other is fermentation. Let's look at ATP first. ATP stands for adenosine triphosphate. It's the major form of energy, as we said, used by cells of the body. It contains an adenosine part and 3 phosphate groups. When the third phosphate group is removed, it leaves a molecule called adenosine diphosphate, 2 phosphates. Energy is made available for use by the cells. ATP is constantly recycled between ATP and ADP. Where does the ATP come from? To understand that, we need to understand a little about oxidation reduction. The energy comes from any oxidizable substance. Usually it's an organic compound. We call it food. It requires oxidation-reduction reactions. Notice we use the phrase oxidation reduction. Oxidations release energy. They always release the energy in the form of electrons. Usually the energy is released as a hydrogen atom, because remember, hydrogen has 1 electron, and so the hydrogen is the carrier for the electron. Sometimes an oxidation occurs when a substance gains oxygen. Reduction, on the other hand, takes energy, always in the form of electrons. Sometimes it gains hydrogen, and sometimes it releases oxygen. An oxidation and a reduction must happen together. The substance being oxidized is a reducing agent because it's needed for the other to be reduced. The substance being reduced is an oxidizing agent. It's needed for the other to be oxidized. It often involves oxidation and reduction of permanent molecules. These are part of the machinery of a cell. Coenzymes, for example, these are usually in solution in the cell. They release or take hydrogen when they're oxidized or reduced. Examples include NAD and NADH2. These generate roughly 2 ATPs each. Another example is FAD and FADH2. They generate roughly 2 ATPs each. By the way, NAD stands for nicotinamide dinucleotide, and FAD is flavonoadenine dinucleotide. Cytochromes are another permanent molecule. These are attached to the mitochondrial inner membrane, and they're oxidized and reduced by releasing or taking electrons. Which is the reduced state, NAD or NADH2? Well, it's the NADH2 because it has gained extra hydrogens. And by the way, technically it's not really NADH2, it's NADH, and the second hydrogen is sort of floating around in the medium. But we'll use the symbol NADH2. Well, now we can look at cellular respiration. It's the major source of energy. It's a complex process in which substances are oxidized one step at a time, and we call it cellular respiration. Several characteristics of cellular respiration. One is purpose is to generate ATP. It's an aerobic process, that is, it requires oxygen. A summary reaction of cellular respiration goes like this. C6H12O6 plus O2 gives CO2 plus H2O plus energy. That is, glucose plus oxygen gives carbon dioxide plus water plus energy in the form of ATP. The rate of oxidation varies. For glucose, 120 milligrams per minute. Fat, 60 milligrams per minute. So glucose is a faster source of energy, so to speak. 75% of all metabolic energy is converted to heat. There are three parts of cellular respiration. We'll detail them in the next few slides. One part is called glycolysis. The second part is the Krebs cycle, or the citric acid cycle. And the third is the electron transport system, or chain. Glycolysis alone will generate roughly 2 ATPs per glucose molecule. The Krebs cycle then generates carbon dioxide and reduced coenzymes. The reduced coenzymes transfer the energy to the electron transport system. The electron transport system requires oxygen and it generates water. The entire process of cellular respiration can generate roughly 38 ATPs per glucose molecule. You see, using glucose molecules as a measure, it helps us to relate the various parts of cellular respiration. Other organic compounds can also be used to generate energy. Fats, a very good source of energy. They're broken down into fatty acids. Proteins can be used under extreme circumstances because the body does not store extra proteins, so proteins would have to come from some of the organs of the body. We'll address this at a later time. Let's look at cellular respiration. Now, yeah, I know the diagram looks terribly complicated, and yet it's a simplification of the process. Well, the arrows represent chemical reactions. We'll come back to those in a moment. It begins, though, with one glucose molecule. And again, we use that because it gives you a useful frame of reference. Remember, glucose is a 6-carbon molecule. Each arrow represents many reactions, usually. Curved arrows are a visual way of showing a reaction of 2 or more substances. In this case, glucose 6-phosphate plus ADP gives PGAL plus ADP. The arrows indicate the direction of the reaction. Some of the reactions are reversible, so I show that with 2 arrowheads. Now, that does not necessarily mean it's the same reaction. It may be a process involving different reactions that achieve the same result. The major substances are listed, and there are many, many intermediates. For example, glycogen, glucose 6-phosphate, PGAL, pyruvic acid, and acetyl-CoA. Let's look at glycolysis. Remember, the first of the 3 parts of cellular respiration. Glycolysis can be defined as the breakdown of glucose to pyruvic acid. It's often called pyruvate because it exists as the ionic form. It begins with one glucose. So what you see in this scheme starts with one glucose. Since glucose is 6 carbons, it may split into 3-carbon compounds, hence the number 2 in front of many of the other substances. Many of the steps are oxidations. In glycolysis, 2 ATPs are added and the result is 4 ATPs are produced for a net of 2 ATPs. In the first step, one phosphate group is transferred from ATP to glucose. It creates a molecule called glucose 6-phosphate. And what that literally means is the phosphate group has been attached to the carbon atom number 6. Now, this is not so much an oxidation reaction as it is an activation reaction. You see, the glucose must be activated in order to stimulate it to release energy. Consider gasoline. If you had a bucket of gasoline, you know it contains a lot of energy. It doesn't just magically release the energy, but you have to add a little bit of energy to release its energy. What do? Simple. Strike a match and drop it in the bucket of gasoline. Don't try that. In many steps, the glucose 6-phosphate is changed to 2 PGALs. These are 3-carbon compounds. In this process, another ATP was required for activation. In many steps, the 2 PGALs are oxidized to 2 pyruvic acids. 2 NADs are reduced to 2 NADH2s. The generation of 4 ATPs by any substance is called substrate-level phosphorylation. Glycolysis to Krebs cycle. This is an intermediate step in which the pyruvic acid is transported into the mitochondria, oxidized to acetyl-CoA in a rather complex process to produce the acetyl-CoA. In this step, 2 NADs are reduced to 2 NADH2s. 2 carbon dioxides are generated. Now about the Krebs cycle. We can define Krebs cycle as the oxidation of acetyl-CoA. It begins with acetyl-CoA. This has come from carbohydrates, fats and other lipids, and proteins. Acetyl-CoA is a 2-carbon compound. Each glucose has resulted in 2 Krebs cycles. So each acetyl-CoA combines with a 4-carbon compound to form a 6-carbon compound. This is citric acid, the name used sometimes for the Krebs cycle. Oranges produce a lot of citric acid. 2 successive carboxylations occur, resulting in a total of 4 carbon dioxide plus 4 carbon compounds. Reduced coenzymes are generated by oxidation of the organic compounds in many steps. 2 FADs are reduced to 2 FADH2s. 6 NADs are reduced to 6 NADH2s. 2 ATPs are generated. Then we have the third part of cellular respiration, the electron transport system. This is composed of a series of cytochromes and complex proteins located, as we said, on the inner membrane of the mitochondria. The cytochromes, I'll abbreviate them CYT, constantly alternate between an oxidized state, I've got the O in parentheses, and a reduced state with the R in parentheses. In so doing, they pass electrons from one cytochrome to the next. The most efficient ATP generating system, but not the only source of ATP, is the electron transport system. It generates ATP by oxidative phosphorylation. This is a little different from the substrate level phosphorylation that occurs in glycolysis. You remember that the molecules that enzymes act on are called the substrate molecules. And so in glycolysis, it's the substrate that releases the ATP. In the electron transport system, it's a little bit different. It needs a continual source of electrons and hydrogen. This is supplied by the reduced coenzymes. NADH2, and FADH2. Each NADH2 generates approximately 3 ATPs because it enters the cycle at a little different point. Each FADH2, on the other hand, generates approximately 2 ATPs of energy because it enters the cycle at a little different point. It requires a final electron acceptor. The final electron acceptor is oxygen. The final electron acceptor accepts electrons and hydrogens. The final electron acceptor is reduced to water. Now, notice what's happening. Electrons flow from left to right, and as they are transferred from one cytochrome to the next, energy is made available to generate ATP. Each cytochrome starts off in the oxidized state. When it accepts electrons, it becomes the reduced state. Well, it has to then restore to the oxidized state before it can accept more electrons. So if you look at the last cytochrome, it is reduced. When it accepted the electrons, it turns around and gives up its electrons and becomes reoxidized. But something has to take those electrons from the last cytochrome. That's the role of the last acceptor, or the final electron acceptor. The generation of ATPs, as I mentioned, is called oxidative phosphorylation. Let's summarize cellular respiration. 2 ATPs are generated directly from glycolysis. 2 are generated directly from the Krebs cycle. These would be the substrate level phosphorylations. 10 ADH2s are generated. These are the reduced form of the coenzymes. 2 come from the glycolysis step, and 2 from the step between pyruvic acid and acetyl-CoA, and 6 from the Krebs cycle. 2 FADH2s. These are in the reduced form as well. These coenzymes are generated from the Krebs cycle. Each NADH2 is transformed into roughly 3 ATPs for a total of 30. Each FADH2 is transformed into roughly 2 ATPs for a total of 4. Therefore, the total from the reduced coenzymes is 34 ATPs worth of energy. Add the 34 from the reduced coenzymes and the 4 produced directly from glycolysis and the Krebs cycle, and the total is 38 ATPs produced from each glucose molecule. The cell uses less because each NADH2 generates less than 3 ATPs, and some of the ATP is needed to transport the pyruvic acid into the mitochondria. Fermentation is the other form of energy generation. It's an alternate source of energy. It's used in particular by skeletal muscles as well as other tissues. It's an anaerobic process, that is, it does not require oxygen. In fermentation, 1 glucose is transformed into 2 pyruvic acids. The 2 pyruvic acids then are changed into 2 lactic acids. It generates a net of 2 ATPs. Now remember, even though the diagram shows 4, 2 of those were used to pay back the cell for the 2 that were used to activate glucose. It generates energy quickly because there are fewer steps involved. So it's useful when tissues have an oxygen deficiency and yet they need to generate energy fast. Skeletal muscles use this source during strenuous exercise. Energy is measured in calories with a small c, or kilocalories with a large C. Recommended average daily calorie intake in males is roughly between 2,800 and 3,000 kilocalories. females roughly 2,000 to 2,400 kilocalories. Calorie output in a sedentary adult includes the BMR and SDA. We'll describe these in a minute. That's roughly 75% of the calories that are used. Thermogenesis, generating heat, 7% of the calories. Physical activity at rest, perhaps 18% of the calories that are used. The metabolic rate is the energy expended in a given period of time. The total energy released includes the work done, plus energy put into storage, plus the heat produced. To measure metabolic rate, direct measurement may be used. A calorimeter would measure the heat released. For example, a person might be put into a device, into a box that carefully measures the temperature or the heat that person generates. Indirect measurements are more common, like the oxygen consumed. 1 liter of oxygen amounts to about 4.8 kilocalories of heat. 3 components of metabolic rates are basal metabolic rate, BMR. This is the minimum energy needed for survival. Metabolic rate is measured under standard conditions, post-absorptive state, you'll hear about that in a moment, lying down at rest, constant room temperature of perhaps 68 to 77 degrees. This amounts to roughly 60% of the calories used. 1,400 to 1,750 kilocalories per day. This would be 20 to 25 kilocalories per kilogram of body weight per day. 40% supplies the central nervous system needs. The central nervous system uses something like 125 to 150 grams per day of glucose. 20 to 30% of the basal metabolic rate supplies the skeletal muscle needs. 30 to 40% supplies the other needs of the body. Specific dynamic action, SDA, also referred to as food-induced thermogenesis, is the energy required to digest and absorb food. Roughly 10% of the calories used are involved in SDA. Physical activity may increase energy expenditures 15 times. And that's the end of part D. The first part, you're now ready to progress on to the second part.